welcome to A Moment of Science, please. I'm Aghia.
And I'm Mohammad.
And we are your hosts for today's episode. As you may already know, A Moment of Science, Please is a student-run podcast based at University of Toronto, which highlights, or at least tries to highlight, some of the fascinating scientific research we have going on here. And today, we'd like to welcome Dr. Tropepe, a professor at the University of Toronto's Department of Cell and Systems Biology, and a vice dean research in the Faculty of Art and Science. Welcome, Dr. Tropepe.
Thank you very much. Happy to be here.
All right, so let's begin with some introduction. I was wondering if you could please introduce yourself, tell our audience a bit about your background and education.
Sure, happy to do that, and good afternoon, everyone. I guess I'll start with my undergrad. I did my undergraduate degree at McMaster University it was a combined honors in biology and psychology. And it was, I think at the time, the closest thing they had to a neuroscience program. And I was quite fascinated with the topic. And so I did that for 4 years and got really interested in neurodevelopment generally as a topic, as a field, and did my PhD in the Department of Anatomy and Cell Biology in the Faculty of Medicine at the University of Toronto, and also completed my degree as part of the graduate collaborative program of developmental biology at the university. And there I studied the role of neural stem cells in early development of the brain and the retina in mice as an animal model system. And also tried to understand how neural stem cells function in mature brain tissue and retinal tissue as well, using that animal model. And got really interested in some of the molecular basis of stem cell function in the nervous system more broadly, and wanted to learn a little bit more in sort of underlying molecular aspects of the behavior of these cells, in particular in early development. And so I decided to pursue a postdoctoral fellowship at the Whitehead Institute as part of the MIT in Boston, Massachusetts, and Cambridge rather. And there, as a postdoctoral fellow, I was able to tackle some projects related to early nervous system development using different animal model systems, in this case, both zebrafish and Xenopus laevis frogs. And those animal model systems are pretty fascinating to study, very early stages of neurodevelopment, because their cellular and molecular processes are so very well conserved among all vertebrates, including mammals. And so I had an opportunity there to apply more molecular and genetic techniques to understand the behavior of these cells. And that got me really excited about doing this more or less as a career when I was then recruited back to the University of Toronto in the Department of Cell and Systems Biology. And I've been there ever since. My lab studies has been for many years now studying neurogenesis, which is the term that's used to kind of think about the production of new cells in nervous system tissue. And we're interested in neurogenesis throughout life, really. And we study it in a variety of different contexts. We study it in very early development to try to understand how you build brain tissue or how you build the retina. These are very complex tissues with a lot of different cell types. And we're trying to understand how stem cells make those decisions to generate all that diversity as the tissue is growing at the same time. We're also interested in the plasticity of this process. And as it turns out, in particular in animals that at very early stages of their lives have to fend off predators and have to find food on their own, they're typically tiny, and they're still growing their neural tissue significantly, but at the same time, it has to be functional because they have to sense the environment and respond to it. And so how do you continue to build a brain where it has to also be working at the same time. And so we're quite interested in the way that activity then lends itself to modulating the process of neurogenesis. And then finally, we're quite interested in regeneration. And we continue to use zebrafish as an animal model in this work. And there we're focusing our attention on the retina, which is a really beautiful system to try to understand how, after damaging the retina, you can recreate those cells through the activation of stem cells. And so that's what my lab has currently been up to, trying to understand basic biology, but also relevance to disease and hopefully to repair damaged tissue.
So as you mentioned, a lot of your research is dedicated to neurogenesis, especially when it comes to visual development. So I guess you can answer this, but like to reiterate, what got you interested more specifically in neurogenesis?
Yeah. So to me, one of the fascinating questions around neurogenesis, as I was alluding to, is how do you generate diversity? The nervous system has billions of cells, but it has dozens and dozens and dozens of cell types. And in fact, if you just think about the retina, there are broad categories of cells that everyone would be familiar with, like photoreceptors. Rods and cones are the two main types of photoreceptors, and ganglion cells are those larger projection neurons that take the information that's being processed in your retina and send it back to the brain so that the brain can then further process that visual information. But even if you look at the retina, there's 6 or 7 main categories of cell types. Within each of those categories, there are many subtypes. And it's seemingly endless, the number of the diverse number of cell types. And so if you have a retinal stem cell, how does the retinal stem cell know to generate these different types of cells? And we've also learned by lots of research that has been happening over many years now, that the stem cells generate these cells, they kind of in a balanced way. On the one hand, they can be very flexible and generate them almost seemingly at random, or in what's often referred to as a stochastic process. But on the other hand, they generate them on a schedule. They tend to make ganglion cells first, and they tend to make rod photoreceptors and other cell types near the end of this process of generating the cells. So there's a bit of timing, there's a bit of flexibility, and we want to know at the molecular level, how do you generate this? How do you control this process? And in trying to understand this by manipulating different kinds of genes that we think, or we have some hint might be involved, and then try to study it in the actual eye itself and manipulate the gene and see the outcome. What happens to the generation of photoreceptors or ganglion cells or other cell types, to try to learn about how stem cells make these decisions to produce the diversity. So for me, that's one of the really key questions in understanding early neural development. But the other reason that we study this, that I'm quite fascinated in this, is that oftentimes these stem cells are very active early when you're building the tissue, but then they go quiescent. And it's true in the nervous system of mammals that most of that neural tissue, once it's generated, it cannot be regenerated. So what we also are trying to understand is, why do the stem cells turn off? Why don't they keep on having that ability to make new cells when we need them to? And there is something about either the inherent program, the genetic program of those cells, or it could also be the environment that is changing, the microenvironment that those cells are in. And if it's changing, that might lead them to sort of go in a state of quiescence. And so we're trying to understand these factors so that we can manipulate these stem cells to regenerate tissue, ideally on demand. We're not quite there yet at all, but just trying to understand the molecular basis of that regeneration process. So those are the main the main areas more specifically that we're really interested in the retina.
Yeah, well, thank you so much for such a great answer. You were talking about retina. I was like, yeah, I know what that is. So, uh, to build on that question and get a little bit more specific, I know that in one of your most recent papers, you talk about a gene that is related to the Usher syndrome. So
Yeah.
I was just wondering if you could like tell us and our audience about this condition and why you chose to investigate it, if there's any specific reason.
Yeah, and we really came about this question about Usher syndrome almost from the back door. And I'll sort of describe what I mean by that. So Usher syndrome is a developmental disorder where the children are born with typically with severe hearing loss right from birth. And they also gradually lose their vision. And so it's sensory neural is kind of a catch phrase that's often used for these types of disorders where it impacts their ability to sense either sound or light in those specialized sensory organs that we have in the inner ear or the retina in this case. And Usher syndrome, the children are born typically, I mean, there's different categories of Usher syndrome, and they're the ones that are the more severe category and those that are slightly less severe. All of them have pretty substantive hearing impairment. And in the more severe case, they're born deaf. Kids are born deaf. With respect to vision, in the most severe cases, the vision starts to become detrimental at very early stages, after the first few years of life, almost toddlers, or within 5 to 10 years or so, they start to lose their vision. Otherwise, in the less severe cases, the vision loss happens as teenagers and sometimes into your 20s or 30s in the least severe cases. It's an interesting question as to why vision loss is so protracted over time, but the hearing loss is very early. And many researchers around the world are trying to understand those differences. There's a really good understanding of why you have the hearing loss, and different genes have been associated with Usher syndrome. And if you consider all the different genes, maybe probably a dozen separate genes that have been associated with this disorder. And when you think about the genes and what they encode, these genes that are involved in this disease typically encode a protein. And as it turns out, each of these genes that have been associated with the disorder encode a protein that works together in a large complex molecularly inside the cell. And that complex in the hair cells of the inner ear, that complex is important to control the structure of those hair cells. And they call them hair cells because when you look at them under a microscope, they have these little microvilli that stick up like little hairs. And those hairs are all interconnected with each other so that they tend to move in unison in any one cell. And then there's a whole bunch of them side by side. And as the sound waves come in, they bend those hair cells. And as the hair cells bend, they open up these molecular channels in their membranes to send a signal inside the cell that eventually gives that signal to the nerve that sends it to the brain. And so they can then, depending on the type of signal, it could be high-pitched sounds or low-pitched sounds, and they can essentially hear that incoming information. What happens in Usher syndrome is that, like I said, those proteins that are associated in the complex, they're important for these connections. When they break down, the hair cells are kind of splayed and they don't kind of move as they should, and then the signal doesn't get created. The same kinds of genes are also associated with the vision loss. And it's unclear how that same molecular complex of proteins works in the retina. And in particular, it's been found that they're present in the photoreceptors. But the photoreceptors don't necessarily need that kind of movement in response to sound waves. Of course, they process the information differently, and they use photons of light that have to activate proteins to send the signal through the photoreceptor to eventually get to the brain. And so what we were quite interested in is the question around structure, right? Because when photoreceptors are born, and of course this gets back to the neurogenesis question, we were interested in how do you make a photoreceptor? When you make a photoreceptor, it starts off as a more simple looking cell type, but photoreceptors are very complicated kinds of cells. They grow this outer segment that has these like hundreds and hundreds of layers of membrane on that outer segment, and packed in there are the proteins important for processing the photons of light. But how do you make that outer membrane? And so we were interested in this question, and we came across one of these genes that is associated with Usher syndrome called protocoherin 15, or PCDH15. And we learned that in the inner ear, that gene is critical to maintain the structure. But we didn't really understand how it worked in the retina. And so we thought we can gain some insight into how to make that beautiful structure of the photoreceptor if we manipulated this gene. So we did. And we created, using Zebrafish, we used a CRISPR-Cas9 technology. It's a gene editing technology where we could go into the genome of these animals and make a mutation in this particular gene. And we did, and the outcome was that they started to build their retina completely normally. And at some point in time, what we noticed was that the photoreceptor, especially the outer segment, was not being generated properly. In fact, it was very, very abnormal, and it started to even detach from the rest of the cell body. And so this was what we think is the first real evidence of a structural malformation in the development of the photoreceptor that could then be associated to why you get the vision loss in Usher syndrome over time. Because initially the photoreceptors are being born and they're being generated, but they don't maintain the structure without this key protein in the complex. And then that leads them to lose their ability to maintain the structure very early on. And in this particular case, it's quite a severe phenotype. And we're now trying to understand what the impact that would have on the actual vision of the animals, and we're trying to test that. So that's how we came into the Usher syndrome field, is because we were interested in that, in photoreceptor development, and from a structural perspective, and came across this, what we think is a really nice now animal model to try to understand the vision part of the disease in Usher syndrome.
So we noticed from your research, and also you said it yourself, that you use zebrafish as a model organism. So I was wondering if you could tell our audience why you decided to go with zebrafish and what's so special about it.
Yeah. Well, I mentioned that I started using this animal model as a postdoc, and I'll never forget in one of my postdoc interviews, at that time, I knew I was interested in using it, but only because I was reading papers. I actually, at that time, had never even seen a zebrafish embryo, for example. And in my interview, I asked one of the people in the lab I was interviewing with and said, can I see one of those embryos under the microscope? And they just showed me these embryos, and they're just beautiful. And at these early embryonic stages, they're completely transparent. So you can actually, and what I saw for the first time was a couple of blood vessels. You can see the sort of wall of everything looks black and white, right? Just under a regular stereo microscope. You see these blood vessel walls, and I saw these little blood cells moving through, and the heart was beating as they were moving through. And it looked like you were in a helicopter over Highway 401. And you have all these roads and all the cars that are moving. And that's the impression that I got. Anyways, the coolest thing ever. So I wanted to study, I wanted to use zebrafish. But we have several reasons why we use it. First, for that reason, in early development, you can see things through these animals because they're transparent. So you can do live imaging of these of early developmental processes. So in fact, I have a student in my lab who's looking at the retina, and very early stages of development. She can video record the behavior of the cells in real time, because we can make transgenic animals that express fluorescent proteins, and we can capture the image and we can watch them over many hours. And you can see individual cells divide and make their daughter cells. And so if you're interested in neurogenesis, this is a beautiful system to study it in sort of looking at the real tissue. So that's one reason. Another reason is with respect to the retina and the brain, they continually produce new neurons throughout life in different parts of these structures. And so in the case of the retina, right at the periphery of the retina, you imagine this retina as a big cup, a three-dimensional cup. And right at what would be the rim of the cup is a rim of stem cells. And they make new cells, and they just contribute more and more cells to the rest of the retina as it's growing. And so we can study the process of neurogenesis, not just in the early embryo, but much later stages of life as well. And because we think that neurogenesis has some fundamental mechanisms that are consistent throughout all these ages, but as you age, some of those mechanisms change. And we'd love to know what changes with age in terms of the molecular basis of neurogenesis. And then the third and final reason is because of regeneration. So in these animals, even as adult fish, if you make a lesion in the retina or the brain or the spinal cord, after about 3 to 4 weeks, all the cells come back and all the function returns. And we would love to know how this works in this little tiny fish that you can get at a pet store, because it doesn't work that way for humans. There are so many fundamental similarities in the molecular programs of the stem cells in fish and in humans, but clearly there's some differences as well. And so we want to understand what those differences are, because we want to be able to then tap into a way to reactivate those stem cells in a human to be able to initiate or facilitate regeneration. Because if a fish can do it, we think that a human could do it as well, but we just need to understand the molecular processes.
Wow, thank you so much for sharing all that. You know, one of the purposes that we started this podcast was to show our audience that research is way cooler than just like, you know, reading papers. And you definitely made it sound like that. So thank you so much. And you know, just follow up on the comment that I made. So far we discussed your research career as a researchers, but we were also wondering if you could talk about your responsibilities as a vice dean of research and how they differ from the responsibilities of a typical researcher.
Yeah, sure. Happy to do that. So my administrative role as vice dean research in arts and science, I mean, it does have some parallels, and maybe I can highlight a few, but it's also a very intriguing opportunity for me that came up to learn a little bit more about research from the institutional perspective. And so as a professor at the university in the field that I am in, I focus a lot on my lab, members of my lab, the students and postdocs that contribute to the research, and other staff members that contribute to the research that we're doing. And I have to apply for grants to get funding to do this work, so I can buy equipment, so I can pay salaries for the fantastic people that are contributing to the work, and to make new discoveries. And that's an essential ingredient to my passion for doing this work, because I wouldn't be able to do it otherwise. And you know, arts and science supports this kind of research. I'm a member of a department that's part of arts and science. And there are ways that the faculty supports this kind of research that would be impossible for me to do otherwise. Space, They have the ability to contribute to supporting individuals that will allow me to manage my grants, that will help me to apply or to develop partnerships with things that would be very difficult for an individual to do on their own. But having the support for from this great institution behind you, people get interested. Say, oh, well, a professor from the University of Toronto, well, that's a great university. It's got a great reputation. Maybe we would want to engage and develop partnerships or provide grant funding, and having the expertise, the individual expertise to help support that. So in my role as Vice Dean for research, I look at it from how do we, as an institution, help support the great scholars that we have in all the various disciplines. And the one thing that I'm learning about in this role is learning a lot more about the humanities and the social sciences and other aspects of science that I didn't really know much about as a developmental neurobiologist, very focused in my own world. And to get a sense of the variety, the diversity of scholarly excellence in all of these areas represented in arts and science is just fantastic. And I learned a lot about how to support scholars in these other areas and how we do this support in areas that are more similar to my field, because there's a lot of interesting cross-fertilization on how you can do that and how you could implement these kinds of supports for research. And so this role lets me look at research from a completely different lens than the individual principal investigator who's doing the work on the ground. And so far it's been a pretty fascinating experience.
So the next question is something that maybe our audience are very interested in knowing about, and that is, what tips or advice do you have for students who are interested in pursuing a career in research?
Oh, yeah. I think it almost sounds cliche, but is to follow your passion. And I say that because I've now had the great benefit of mentoring a lot of different students over many years, graduate and undergraduate. And the students that really benefit from the experience in the lab, and everyone comes in, there's a lot of common experiences, like my experiment didn't work, I forgot to do this, or something broke, or it's the day-to-day grind of doing research like this, which is lots of failure. But the students that go into it thinking, this is really interesting work, very passionate to understand how this works, and passionate to learn new techniques or new ways of thinking about the problem that I'm trying to solve. And the kind of person who wants to think about other information that's out there and integrate that information into their area of interest, and kind of take that, blend it with their own personality and creativity, and come out the other end with some interesting new ideas of how to pursue a research question. Even if they have these setbacks. So they have that resiliency to say, oh, another setback, something didn't work, but they continue to push forward because they have that drive and that passion. Students that showed that were very, very successful in pursuing careers in research. And to be honest, you kind of need it, because you're going to write a lot of grants as a researcher that never get funded. It can be very depressing and very hard. But you need to pick up the pieces and keep going, because there's another opportunity coming up around the corner. And so if you have that kind of personality and you have this joy of discovery, then I think research could be for you as a career choice. Other people choose research for different reasons. They have a very particular career in mind, and they know that research could be a stepping stone. There are particular skills that you learn in research that they want to learn these skills, and they want to adapt them to a future career where they're going to use them in a slightly different way. And I think that's fantastic as well. And I think that's one thing about research that can open a lot of different doors, is learning to to be critical in the sort of very productive critical way, to be analytical, to be creative with your ideas, and to learn to communicate those ideas. You learn those things as a researcher. And so if you have those desires, and if you think you might like that, then maybe trying research would be great. And there's programs you can do, like a master's program that's short-term, very focused, and you can use that for other pursuits, but just gaining those skills. If none of this seems appealing to you, then you definitely should not do research. Most importantly, I think resilience is one of the most important attributes of a really great researcher. So, yeah.
So by tradition, we always ask our guest speakers to tell us about their favorite scientist and why that's the case. So if you could tell us about yours.
Yes, favorite scientist. That's a hard question, I have to say. So I couldn't choose just one. So I'm going to tell you a historical one and then more contemporary pair of scientists that whose work I just find completely fascinating. So the historical one is Santiago Ramon y Cajal, who was a Spanish neuroanatomist, actually won the Nobel Prize early 1900s. I can't remember, 1905, 1906, something like that. And Cajal and is one of his contemporaries, actually, Camilo Golgi, they were looking at brain tissue basically for the first time under a microscope where they can actually see cells. Prior to that, they would be looking at brain tissue from lots of different animals and from humans. And it just looked like a light gray, beige, nothing. No detail, couldn't see anything. And so what Cajal did with his contemporaries is they figured out ways of using different chemicals to stain the tissue. And all of a sudden they could see the beautiful cells with all that complex intertwined structure. And what Cajal figured out in a very painstaking way, and he was drawing beautiful pictures of these cells because there was no imaging that they could do at the time. But what he figured out in his brilliance is that it's not a complete ball of mass of cells. They're actually individual cells. And they communicate through their little tips, which later we learned were synapses. And so he can see all of that in his painstaking work at that time. And so for me, that was like mind-blowing and really contributed so much. And in fact, researchers in my field are always quoting Cajal and showing images of Cajal's work because it was so prescient in terms of, guiding the future of this field for hundreds and thousands of researchers over many decades. So that's my kind of historical example of someone who I find, whose work I find fascinating. The other is more contemporary, and it's Richard Axel and Linda Buck. And they also won the Nobel Prize just several years ago. They figured out that the genome encodes genes that produce proteins that are for olfaction, for the sense of smell. So individual genes encode information for individual types of olfactory receptors. And what their team, plus others at the time, figured out is that each of those olfactory receptors, if one gene gets expressed in one type of olfactory receptor cell inside your nose, in the epithelium inside your nose. And they also figured out that that one cell then makes a projection from your nose up through the skull and into your olfactory bulb in the brain. Only one projection. And so now you have different olfactory receptor cells expressing only a single olfactory receptor gene that is sensitive to only one molecule that you smell, and it's projecting to the brain in a almost topographical pattern. And so that to me was a fascinating set of work, like seminal work, to try to understand how the brain builds the connections to understand the environment. And that to me was just like breathtaking work that they did, that their teams, and they also very much deserve the Nobel Prize for that. So those are two general examples.
Well, thank you. We know that this question usually is not easy to answer, but you did a great job of answering it. And with that, I want to thank you for coming here today. And it was such a pleasure talking with you. You definitely are doing very, very fascinating research, and I hope that our audience find it interesting as well. Yeah, just to our audience, this concludes our podcast. If you liked it, please share it, subscribe, and follow us. Thank you so much.